This is part 4 of the Chapter 5 lecture notes, and we left off talking about how paramecia are freshwater protozoans or protista, which a lot of times organisms that live in fresh water tend to take on water. They tend to gain water because fresh water is hypotonic compared to their cytosol, or the fluid inside of their bodies, inside of their cells. A paramecium is a single-celled or unicellular organism. So its cytosol has a higher concentration than the fresh water. And down at the bottom, it says marine invertebrates. And of course, marine refers to ocean, salt water. Invertebrates have an invertebrate that lives in the ocean would be like an octopus or just anything without a backbone. So an octopus or a clam or a crab. Marine invertebrates have internal salt concentrations that match their environment. So if they match their environment, that means the ocean water is isotonic compared to the salt concentration inside their cells. And I can relate to this because in my family, we do a lot of fishing and we do freshwater and marine fishing. We go to the coast and fish a lot, and and if I cook like a catfish or a fish that I've caught, in the river or a lake, a lot of times I will put it in salt water and let it sit for a little while before I cook it. But I never do that with a fish that I've caught in the ocean because it's already salty enough. So, and that's the reason why. Fishes excrete diluted urine to get rid of excess water or salts. So that's one way they help regulate their salt concentrations. If they're getting rid of excess water, that means that their salt concentration is to, let me think. If they're getting rid of excess water, that means their salt concentration is not as high as it is in the ocean. But if they're excreting salt in their urine, that means that their salt concentration is higher than it is in the ocean. And for organisms like us, we have kidneys, so there are receptors in the brain that monitor the salt concentration or solute concentrations in our blood. They're called osmoreceptors for osmosis. And these osmoreceptors will cause hormones to be released that affect kidney function, that affect whether or not we retain water or we release more water when we urinate. So it affects the volume of urine and the concentration of the urine that we excrete. Now, so far when we've talked about transport across the plasma membrane, it's been passive. Now we're going to discuss active transport, which has two differences between passive from passive transport. One is the direction that the molecules move. Molecules are going to move in active transport. They're going to move against their concentration gradient. Instead of from high to low concentration, they're going to move from low to high concentration. And another thing in active transport is that energy is required. Energy is always required for active transport. There are two types of active transport. There is primary active transport, where the energy that's needed is provided by ATP. And then there is secondary active transport, where an electrochemical gradient provides the energy. And that means there's a difference not only in the concentration of solute on either side of the plasma membrane, but also a difference in the electrical charge. So electrochemical refers to a difference in the electrical charge as well as a difference in concentration across the plasma membrane. So electrochemical gradients is the subject of this next slide. They result from the combined effect of concentration gradients and electrical gradients. The electrical gradient comes from the fact that the cytoplasm contains more negatively charged molecules, more negative ions and proteins, than the fluid outside the cell. And that is critical for proper cell functioning. It makes the voltage on the inside of the cell negative. Sometimes, like in a muscle cell or a nerve cell, that voltage is negative 70 millivolts. Sometimes it's different, but it's some negative number. And that comes from the fact that there are less positive ions and more negative ions inside the cell. And there are also these large proteins that stay in the cell that aren't allowed to leave the cell because they're too big, that are negatively charged. And these keep the inside of the cell negatively charged compared to the outside. So the net charge inside the cell is negative, and the net charge outside the cell is positive. And that's the electrical gradient. Now, the chemical gradient results from the concentrations of the solutes. So the chemical gradient for potassium, for example, potassium is going to be high inside the cell and low outside the cell. And then sodium is going to be high outside the cell and low inside the cell. And so that's the concentration gradient part. That's the chemical part of the electrochemical gradient. So combined, the electrochemical gradient will determine which direction, like let's go back to this page. So let's say that there is a protein somewhere in the plasma membrane that's going to allow sodium to just diffuse passively. Let's say it's a carrier protein, and it's going to allow passive transport or passive diffusion of sodium. So sodium has a positive charge, and it also has a high concentration outside the cell. So for two reasons, it's going to move in if it's allowed to diffuse. It's going to move in because its concentration is high outside the cell. And it's also going to move in because positive is going to be attracted to negative. Because sodium is positive, it's going to move into the cell where the charge is primarily negative, is overall negative. So because of the charge and the concentration gradient of sodium, sodium will move in if it's allowed to, if it has a carrier or a channel protein that it can passively move across the plasma membrane through. Now, active transport involves carrier proteins, except the only difference is they require ATP energy, or they require energy in some form, and they are pumping molecules from their low to their high concentration. So a uniporter carrier protein is just transporting one molecule, molecule A, from its low concentration outside the cell to high inside the cell. So a uniporter carries one molecular ion. A symporter transports two different molecules or ions in the same direction. An antiporter, like the sodium-potassium pump that we're going to talk about, transports two different molecules in different directions. In primary active transport, the energy comes from ATP, and the molecules are pumped against their concentration gradients. An example of primary active transport is the sodium-potassium pump. The sodium-potassium pump is necessary to maintain the proper resting membrane potential for cells. In other words, to help maintain that the inside of the cell is negative and the outside of the cell is positive. the sodium potassium, I was about to do an asterisk, I'm sorry, but the sodium potassium pump helps keep the voltage or the electric potential where it needs to be for proper cell functioning. So what it does is it, using the energy of an ATP molecule, it moves 3 sodium ions out and 2 potassium ions in at the same time. And so, question, is this pump a uniporter, symporter, or antiporter? Of course, it's an antiporter. This pump is an electrogenic pump. What do you think that means? Well, it's creating an electrical gradient. I'm going to look that up and get back to you because we're not out of time yet, but I'm going to write it down, electrogenic, and I will let you know for sure what that means, but I'm assuming that it's not a term I've heard before, and I'm assuming it has to do with the fact that it's helping to maintain an electrical voltage across the plasma membrane, helping keep it negative on the inside and positive on the outside. Now let's look at secondary active transport. Secondary active transport uses primary active transport to move a substance against its concentration gradient. So you have to have primary active transport in order to have secondary. All right, so in this case, many amino acids and glucose, which are, both of these are important nutrients. Amino acids come from proteins, glucose comes from carbohydrates. But many amino acids and glucose enter the cell by secondary active transport. So what you can see here is glucose and amino acids are coming in. Let's see. Glucose is, okay, so it looks like maybe sodium is this molecule. I do not like this illustration because the hexagon shape is the shape of glucose. Glucose has a hexagon shape, but they are using the rectangular looking, they look almost like gold bars to represent glucose in this case. And glucose is being moved from its low concentration outside the cell to its high concentration inside the cell. And that is because our cells need glucose. Even if they already have a high concentration of glucose, they need more. More and more glucose means the cell can make more energy. So that's one reason why you need glucose, even if the concentration inside the cell is already high, because glucose is a nutrient. Same thing with amino acids. So what's happening is sodium is coming into the cell because the sodium potassium pump kept the concentration of sodium high outside the cell. So we first have the sodium potassium pump, which is primary active transport. The sodium potassium pump is creating this concentration gradient where sodium is high outside the cell. So when it is allowed to move into the cell through this secondary active transporter, it binds, and then also glucose binds that same transporter. And so they're all moved into the cell at the same time. So it's the primary active transport of sodium out of the cell that provides the energy or the electrochemical gradient that causes sodium to move back in. And in this case, glucose is allowed to kind of take a ride, hitch a ride along with the sodium. And how can this be called active transport when ATP is not used? Because ATP is used indirectly and we're moving a substance against its concentration gradient. It's being moved from low to high concentration. All right, and the next section is bulk transport. Bulk transport means that the material is too large to be transported through the phospholipid bilayer, and it's too large to come in through a transport protein. So it is a type of active transport because it does require energy, but it is the transport of a large amount of material.